Hi, my name is Katie Amberg-Johnson, and this year I'm writing my undergraduate thesis on this project. It's the use of molecular tweezers to investigate a fracture point of sigma 54 core binding domain. So the reason that we're interested in sigma 54 is because it's a protein that's necessary for transcription initiation of certain genes in bacteria. So right now in A, it's in a closed promoter complex, meaning that even though it's associated with RNA polymerase, it still cannot initiate transcription. What needs to happen is a histidine kinase will activate an activator protein, which will go to its active form and will loop around the DNA to contact sigma 54. And after ATP hydrolysis, now the whole enzyme, sigma 54 RNA polymerase, is in open complex form, and that's how it can initiate transcription. So what we're interested is how this happens. How does sigma 54 go from closed to open complex? So looking a little closer, you can see that from residues 1 through 69, that's where it contacts the activator. And residues 69 through 198 is where it contacts RNA polymerase, and we call that the core binding domain. And then these further segments down here, this is where it contacts the DNA. So somehow sigma 54 has to initiate transcription down at this right end after the binding and ATP hydrolysis of the activator on this left end. And that's what we're interested in, what happens there. So there's two ideas about how this could happen. The first is that the activator binding domain of sigma 54 inhibits open complex until it is contacted by the activator and ATP hydrolysis. The second idea is that ATP hydrolysis cycles of the activator cause the activator binding domain of sigma 54 to be pulled through the pore in the activator, which will tug the rest of sigma 54, and as a result, the protein will shift into open complex. And what my project is, it's looking a little deeper into this second hypothesis, and it's testing out this second hypothesis, so keep that in mind. So here is the core binding domain. This is the segment of the protein that I'm working with, and you can see it's comprised of a 3 helical bundle and a 4 helical bundle that is connected by a 10 residue long hinge point right here. And the 4 helical bundle contains most of the contact points to RNA polymerase and is stable, while the 3 helical bundle is not intrinsically stable, and it needs interaction with the 4 helical bundle to fold. The hinge point between the two subdomains is a highly conserved feature of the core binding domain. So these little structural facts propose that the hinge point could be holding the two subdomains together only until there is an applied stress by the activator binding domain, which would induce the conformational change in the core binding domain, which causes polymerase to attain a new state in which it can open DNA and initiate transcription. And that would be also aligned with the second hypothesis. So my project aims to stimulate this proposed tugging by the activator binding domain as a result of the activator on the core RNA polymerase binding domain with the use of molecular tweezers. So here's the setup right here. The protein will be flanked by 2 long DNA handles, and the handles can interact strongly with the beads that are held by a laser. And the laser will pull on the beads, and first the protein will stretch, and then it will unfold. And what we're going to do is measure the number of unfolding events and determine whether the fraction point sort of plays a part in this experiment. So first we have to create the DNA handles, and they're around 500 base pairs each, and each have a thiol group on one end that can bind with the free cysteines at each end of my protein. And the next end, the other end of the DNA handles will be either biotin or digoxygenin. So the reason why we have either biotin or digoxygenin is because they can interact strongly with the beads of the molecular tweezers. And then the next thing you have to do is you have to prepare the protein. So what we do, it's pretty standard. We transform into E. coli Rosetta cells and then induce them to express the protein. The cells are then lysed and pelleted, and the protein is purified on the nickel column. It's pretty standard. So now the next step is you need to attach the DNA handles to the protein, the core binding domain of Sigma54. And this is kind of a tricky step because, as you can see, the protein itself has two free thiol ends, and they will more readily form a bond with themselves intramolecularly than with a separate molecule. And so because of this, you have to protect the ends with DTDP, dithiodipyridine disulfide, so that you get this sort of protected protein, which will then, in the presence of DNA, exchange, and you'll have some product that will be your protein flanked by two DNA handles. And so for my protein, it was a little bit more complicated because it just so happened that the two free ends were unusually close to one another. So I had to take a few extra measures. So we had to denature the protein to separate the ends as far as possible before we actually derivatize with DDP. And then we had to do a buffer exchange with the DNA so that it was out of DTDP, and then immediately put it with the protein and allow it to react overnight. And then we actually purify the protein from the unreacted DNA before setting it up in the molecular tweezers trap. So the idea is that after it's gone through the molecular tweezers, there's two possibilities for what our data could look like. And possibility A over here just shows one rip, and it's this kind of horizontal part of this graph that I drew. And that would indicate that the core binding domain unfolds concertedly. And this data would not support the second hypothesis. So the other possibility, possibility B, is that there are two rips, one for each of the subdomains of the core binding domain. And this would support the hypothesis that physical stress causes conformational changes throughout the molecule to initiate transcription. So this actually would support the second hypothesis. And so that's what, it's either possibility A or possibility B, that's what we're looking for. So I would like to thank my lab head, Dave Wemmer, and the graduate student that I work with, Alex Siegel, and all the other members that have helped me to figure out how I'm going to do this and what I'm going to do. And I'd like to thank you for listening.